But our last medical speaker is Dr. Ken Desantes. He's from the University of Wisconsin in Madison, and his talk today is going to be on MiNiVan trial and the Clarity trial. And unfortunately, he has to do his own intro. I apologize.
That's quite all right, Pat. I think you've already introduced me. So I'd like to think they saved the best speaker for last. I know that's not really the case, but it is a pleasure to be here, my first time speaking at this meeting, and I'm going to talk about the use of molecular targeted radiotherapy to treat children with neuroblastoma. I don't have a slide, but I do not have any conflicts of interest to disclose. So what is molecular targeted radiotherapy? Well, probably everybody in this room is familiar with external beam radiotherapy, where you direct a beam of very high energy particles like protons or electrons directly at the tumor, and it's a very effective way to kill cancer cells. However, you can only treat one or maybe a very limited number of sites at the same time. With molecular targeted radiotherapy, a compound that is selectively taken up by the cancer cells is labeled with a radioisotope and then injected into the bloodstream. So the compound sort of acts as a guided missile, bringing with it a radioactive payload that can seek out the cancer no matter where it is within the body, so you can treat multiple metastatic sites at the same time. So for neuroblastoma, the best example of a molecular targeted agent is MIBG. And this was first described as a treatment for children with relapsed neuroblastoma in 1984. And that's the year I graduated medical school, so this has been around for quite some time. It's mainly been used to treat children who have relapsed or refractory disease. But recently, as many of you are probably aware, the Children's Oncology Group just completed a trial where the MIBG was being incorporated into the initial therapy for children with high-risk disease to see whether it might improve their prognosis. The results of this study are not yet available. So the way this works is the MIBG is labeled with a very large amount of radioactive iodine. So you can deliver a large systemic dose of radiation to the primary tumor, but to all metastatic sites at the same time with relative sparing of normal tissue because the agent is preferentially taken up within the tumor cells. And the way this kills tumor cells is that the cells will directly take the MIBG up with the radioactive iodine into the cell, and that will kill the cell. But there's also something known as the innocent bystander effect that helps with the efficacy of this approach. And that's shown here. So if this is a tumor, and the radioactive symbols represent cells that have taken up the I-131 MIBG, you can see certain cells in the tumor have taken it up and others have not. But the radioactive iodine is firing off these high energy particles in all directions. So neighboring cells can still be killed in this crossfire of radiation. So because the amount of radioactive iodine is so high, to administer this therapy is relatively complicated. It requires a team of very highly trained individuals, shown here. Oh, no, sorry, that's the wrong team. Okay. So it does require the input of nuclear medicine physicians, technicians, pharmacists, pediatric oncologists, nurses. It is a complicated therapy, so not every center in the country is able to administer this compound, but there may be now perhaps 25 or 30 centers that can do it in the United States. This shows how a room would be set up for an MIBG treatment. So most of the radioactive iodine is actually excreted through the urine. So patients have a Foley catheter placed prior to treatment. The Foley catheter drains into a bag that's kept in this lead-lined box that you can see. There's a hole drilled in the bottom of the lead box. A tube connecting the bag runs out of the box through a pump, and then the other end of the tube is directly fastened to the toilet. So every 4 hours, a parent will go into the room, turn on the pump, and the radioactive urine is drained into the toilet. The toilet gets flushed 4 or 5 times, and that's how we get rid of the radioactive waste. So it usually takes about 2, maybe 3 days for the child to get rid of enough radioactivity that they can be discharged from the hospital and go home. So what are the side effects? What's the toxicity of MIBG therapy? In general, it's actually a pretty well-tolerated agent. There can be some nausea because the radioactive iodine can be irritating to the stomach, but it's usually less nausea than what most chemotherapy drugs cause. Sometimes patients will experience transient jaw pain because of salivary gland swelling. But really the biggest side effect typically is boredom, because patients are feeling pretty well. They have to be bedridden for a few days because of the Foley catheter, and we have to figure out ways to keep them entertained. Within the first month after treatment, the main side effect seen is bone marrow suppression. So patients' counts will drop, and they may require red blood cell and or platelet transfusion support. And some patients may need stem cells reinfused in order to help with count recovery. There are some potential late side effects as well. The most common by far is hypothyroidism, and that can occur in about up to 20% of patients treated with MIBG. And then much less commonly, a few other more serious side effects have been reported, including the development of a secondary cancer, especially leukemia. And then some patients take a really long time to recover their platelet counts, even if they've been given their own stem cells back. So how effective is this therapy? Well, I'm showing you here 3 different publications that address that particular question. The first one is actually a meta-analysis of 27 different studies utilizing MIBG therapy, treating over 1,000 patients. Now, many of those patients received MIBG in combination with other agents, but about 200 patients received the MIBG alone. And they noted an overall response rate, so complete responses plus partial responses, of 32%. Another study published by Kate Mathey was a limited institution phase 2 trial, and they treated 164 patients and reported an overall response rate of 36%, with an additional 34% of patients achieving stable disease. And then the University of Pennsylvania published their results, treating 76 patients, and they noted an overall response rate of 30%, with 49% of additional patients achieving stable disease. So just to summarize this, MIBG is actually a pretty effective therapy for children with relapsed or refractory neuroblastoma, with objective responses being seen in up to a third of the patients treated. Many other patients will achieve stabilization of their disease for some period of time, often weeks to many months. And patients who have pain because of bone metastases, often the pain is palliated by the MIBG that again can last for weeks to several months. However, these responses typically are not durable, so unfortunately this is not a curative therapy for relapsed neuroblastoma, and it's not an especially effective way to eliminate metastatic disease to the bone marrow. The role for using MIBG therapy in newly diagnosed high-risk patients has not yet been defined. So a number of investigators have looked at ways to try to make this therapy more effective by combining it with other agents. So for example, the NANT ran a trial where children were randomized to receive MIBG alone or MIBG in combination with chemotherapy, in this case irinotecan and vincristine, or MIBG with another drug called vorinostat, which is a radiation sensitizing agent. And what they noted in this trial was that the children randomized to receive the combination of MIBG and vorinostat had about twice the objective response rate compared to children who received the MIBG alone or the MIBG combined with chemotherapy. Now, one caveat here is you'll note the response rate to MIBG alone is a bit lower than what's been observed in other studies. But be that as it may, it was still a randomized trial, and based on these results, the company that manufactures the MIBG opened a phase 2 study known as the Optimum trial, hoping to achieve additional data combining MIBG with vorinostat to obtain FDA approval to use MIBG as a treatment for neuroblastoma in combination with vorinostat. And this study is very close to completion. So we've adopted a different approach, and that is combining MIBG therapy with immunotherapy. And that's what I've been asked to focus on today. So I'm going to go into a little bit more detail about this trial. And I'd like to actually share with you some of the preclinical data that went into the development of this study, because I think you'll find it interesting. Most of these experiments were run in the laboratories of Dr. Zach Morris, who's one of the radiation oncologists at UW, and Dr. Paul Sundell, who's a pediatric oncologist at UW. So this slide is showing you an animal model of cancer. And the mice are injected in their flank with a tumor, in this case melanoma. The tumors are allowed to grow so they reach a size that you can actually feel the tumors. And then the tumors are irradiated with external beam radiation. And then the tumors are injected with an antibody that recognizes the cancer, an anti-GD2 antibody. But the antibody is also linked to another drug called interleukin-2 or IL-2, which is an immune-stimulating drug. And the combination of the antibody with the immune-stimulating drug is known as an immunocytokine. So after injection with the immunocytokine, the animals are then observed for tumor growth and survival. And these are the results. So if you inject the tumor with an irrelevant antibody, one that doesn't recognize the tumor, shown in the gold or yellow line, the tumors just continue to grow, which is shown in the top graph, which is a tumor growth curve, and the animals all die, which is shown in the bottom graph, a survival curve. However, if you inject the animals with the immunocytokine, or you treat the tumors with radiation, shown in the green and black lines, the tumor growth is delayed somewhat, but these animals eventually mainly die. However, if you combine the radiation with the immunocytokine, shown in the purple curve, now these tumors regress and most of these animals are cured. So there's synergy between the radiation therapy and the immunotherapy. And this experiment shows the same exact phenomenon in a neuroblastoma tumor model. And once again, the animals that were treated with a combination of an anti-tumor antibody as well as radiation therapy had the best results, best therapeutic results, shown in the pink curve. The tumors didn't grow very well, they tended to shrink, and most of those animals were actually cured of their disease. Now, the problem with this model, though, is it doesn't really mimic the clinical situation very well, right? The problem that we have with neuroblastoma is not so much we can't control the primary tumor, it's that we can't get rid of metastatic disease. So to simulate metastatic disease, a different model was developed where animals were injected on both flanks with tumor. So they have a primary tumor on the right side and a secondary tumor representing a metastatic site on the left side. So now what happens if you treat that primary tumor with radiation and the intratumoral immunocytokine injection? Well, if you do that now in the presence of a secondary cancer, the primary tumor continues to grow, which is shown in the dashed green line on the left, and these animals all die, shown in the dashed green line on the right. Now remember, this same therapy was previously curative for most of the animals when that secondary cancer wasn't present. However, if you irradiate that second tumor and treat the primary tumor in the same exact way, now the primary tumor actually shrinks down, shown in the solid green line on the left, and most of these animals are cured, shown on the survival curve on the right. So, what's going on here? There's something about having a secondary cancer, a metastatic disease, that prevented an immune response from being generated to the primary tumor, and those animals therefore died of the cancer. This inhibition caused by the secondary tumor, the metastatic site, could be reversed if you irradiated the secondary cancer or secondary tumor. And that's another example of synergy between radiation and immunotherapy. But what is going on here? Why is that metastatic site causing so many problems and preventing an immune response from developing? So the investigators tried to dissect that, and what they found is shown in this slide. So if you look at the upper left picture labeled A1, this is a primary tumor that's not been treated. You can identify a number of cells shown by these arrows known as regulatory T cells or Tregs, and I think that was just mentioned in the previous talk. So these are immune inhibitory cells. They dampen the immune response. If you irradiate the primary tumor, shown in the bottom picture, labeled A2, you get rid of a lot of these inhibitory cells. You can't find them anymore. Now, if you irradiate the primary tumor, but a secondary tumor is present, you can still detect a lot of those inhibitory cells in the presence of a secondary tumor if it's not treated, and that's shown in the upper right-hand picture labeled A3. So what we think is going on here is that these inhibitory T cells, these Tregs, are present in the secondary cancer, the metastatic site. These cells can gain entrance into the bloodstream, reimplant themselves into the primary tumor, prevent an immune response from developing and allowing the cancer to take off and kill the animals. However, if you irradiate that secondary tumor, you get rid of those T regulatory cells, and now an immune response can be generated within the primary tumor and the animals can be cured. Now, unfortunately, that's not the only mechanism that tumors have to evade an immune attack. There's another mechanism that involves something known as checkpoints, and that's shown on this slide. So T cells, which are one of our immune cells that you've heard about from other lecturers today and yesterday, one of our important immune cells, have activating receptors present on the surface that turn the T cell on. So if, for example, you get infected by influenza, the T cell can recognize cells that have been infected by the influenza virus. T cells become activated and can get rid of the virally infected cells. But T cells also have inhibitory receptors, in this case labeled PD-1. And if the inhibitory receptor sees its corresponding ligand or antigen, it sends a negative or inhibitory signal to the T cell, and that turns it off. So tumors have figured out a way to co-opt this mechanism, and they can express these inhibitory ligands that turn off the T cells and prevent the T cells from generating an immune response against the cancer. Now, over the past maybe 15 to 20 years, drugs have been developed, mainly monoclonal antibodies, that either bind to the inhibitory receptors on the T cells, shown in the red triangle, or they bind to the inhibitory ligands present on the tumor cells, shown as the yellow circles. But either way, they block this inhibitory response from occurring. This allows the immune system to be activated, go in and do its job and kill the cancer. It sort of takes the brakes off the immune system. And this mechanism of action is known as checkpoint blockade. And it turns out this has become a really important tool in our armamentarium to treat cancer, especially adult cancers like melanoma, lung cancer, but it also has proven to be a very important weapon for certain pediatric cancers. Okay, just going back to the laboratory just for a moment. This is another mouse model of neuroblastoma using a neuroblastoma cell line known as NXS2. This is a very poorly immunogenic cell line. In other words, it's very hard to generate an immune response against this particular type of neuroblastoma. So these are animals that have been injected with that cell line. If you treat the animals with molecular targeted radiotherapy, it really doesn't do much. If you treat the animals with checkpoint blockade, it doesn't do much. However, if you combine checkpoint blockade with molecular targeted radiotherapy, shown in the green curve, now the tumor shrinks and most of these animals can be cured. And in the very last preclinical slide I'm going to show you, this is a model that probably most closely resembles the clinical situation. So these mice have a large primary tumor, a smaller secondary tumor, and multiple sites of metastatic disease. And the point I want to make here is if you look at the green bars, this represents the percentage of animals that have been cured with different therapy combinations. And what you can see here is that the therapy that works the best, that cures the most number of animals, is the combination of checkpoint blockade with antibody therapy with molecular targeted radiotherapy. And that really was the rationale that led to this MiNiVan trial being developed. So the MiNiVan trial uses an anti-GD2 antibody. In this case, it's dinutuximab beta. So you've heard a lot about dinutuximab, which is the commercially available antibody to treat neuroblastoma in the United States. Dinutuximab beta is very similar to dinutuximab. It's manufactured a little bit differently, but it's administered very differently. It's administered as a 10-day continuous infusion, and it's given through this device, which is known as an elastomeric pump. It doesn't require any electricity. It's simply a balloon that deflates over time and pushes the antibody into the bloodstream at a constant rate. The antibody is combined with checkpoint blockade, an anti-PD-1 antibody known as nivolumab, and then the molecular targeted radiotherapy is administered by utilizing MIBG. So this slide shows the eligibility for the MiNiVan trial. The main things I want to point out is that patients have to have MIBG avid disease, since MIBG is part of the therapeutic approach, and we don't allow patients to have previously been treated with an anti-PD-1 checkpoint agent, which really isn't much of a problem because these drugs are not commonly used for the treatment of children with relapsed neuroblastoma. The main objective of the trial is to determine the safety and tolerability of combining MIBG therapy with immunotherapy. Secondary objectives are to determine the efficacy of this approach, and there are some biologic corollary studies embedded into the protocol as well. This shows the schema for the trial. So patients first receive 2 doses of I-131 MIBG, given 2 weeks apart. And then 2 weeks after the second MIBG dose, patients are treated with immunotherapy, which is the nivolumab given once every 2 weeks, and the dinutuximab beta infusions given every 6 weeks. The protocol is fairly close to being completed. We expect to meet our accrual goals probably by the end of this calendar year. Okay, I'd like to talk about a different molecular targeted agent known as copper sartate. So this is produced by a company known as Clarity. And there are 8 centers in the United States that are participating in this trial. 8 centers are listed here. So how is copper sartate different from MIBG? Well, MIBG gains entrance into the neuroblastoma cell utilizing something known as the norepinephrine transporter, or NET. The copper sartate gains entrance into the neuroblastoma cell by a very different receptor known as the somatostatin receptor. So if the neuroblastoma has downregulated their norepinephrine transporters and maybe don't take up MIBG very, very well anymore, they still may take up the copper sartate quite well. And this slide shows the comparison between an MIBG scan on the left with a copper sartate scan on the right. The green arrows show lesions, metastatic lesions, that are seen both on the MIBG scan and on the copper sartate scan. The red arrows show lesions that were picked up on the copper sartate scan but were not seen on the MIBG scan. So at least for some patients, it may be that copper sartate is a better imaging agent than MIBG, and potentially a better therapeutic agent as well, although obviously we don't have the data yet. Another advantage of copper sartate, though, over MIBG is that you don't have the same radiation safety precautions as you do with MIBG, so it's an easier therapy to administer. So these are the inclusion and exclusion criterias for that study. And the main thing I want to point out here is that patients who enroll in the study first have a copper sartate scan. It's like a PET scan using copper 64 sartate. And you have to be able to demonstrate at least one lesion, one neuroblastoma site, that takes up the copper sartate to a greater extent than is seen in the liver in order for patients to proceed to the therapeutic phase of the study. And patients can receive multiple infusions of copper 67 sartate, which is used for therapy, provided it's tolerated and there's no evidence of disease progression. So the primary objectives of this study are shown here. It's to determine the maximum tolerated dose of the copper sartate, so it's a phase 1 trial. It's to determine the safety and tolerability of administering copper sartate to patients with relapsed or refractory neuroblastoma, and of course to look for early evidence of anti-tumor efficacy. This is where the trial currently is. I am told that we just most likely completed accrual to the 4th and final cohort. And once this goes through review, then the protocol will move on to a cohort expansion phase where 10 additional patients will be treated at the maximum tolerated dose, which I think will very likely be the dose that's been utilized in cohort 4. And then just for the sake of completeness, I'd like to mention one other molecular targeted agent known as lutetium dotatate. So this is another drug that is taken up by the somatostatin receptor. And there's a phase 1 study that's currently only open in France. The reason I'm mentioning it, though, is that I think there's a reasonable chance this study is going to open in the United States at some time in the near future through the POETIC Consortium. And the POETIC Consortium is another pediatric oncology consortium that runs phase 1 and phase 2 trials for children with cancer. Okay, this is the last slide, and this really has nothing to do with molecular targeted radiotherapy, but it highlights an approach that I've been very interested in over the past 20 years, and that is using haploidentical stem cell transplantation to treat children with relapsed neuroblastoma as well as other cancers. So we've talked a lot about autologous transplants and whether or not they're needed as the initial treatment for neuroblastoma. But what happens in an autologous transplant is you're treating the patient with very high doses of chemotherapy. And the idea is to use the chemotherapy, these high doses, to try to kill as many cancer cells as possible. The patient then receives their own stem cells back just to save them from the effects of the chemotherapy. With a haploidentical transplant, the parent of the child with cancer actually serves as the stem cell donor. And the idea here is to use the parent's immune system to kill their child's cancer. Kind of poetic justice, I think. So this article was published last year, and they treated 68 patients with relapsed or refractory neuroblastoma by haploidentical transplant. And then the patients received immunotherapy starting at 2 months post-transplant. The immunotherapy that they received was dinutuximab beta, which is the same antibody I mentioned that we're using in the MiNiVan trial, and that was given with the first 3 cycles. And then the next 3 cycles, the dinutuximab beta was combined with that interleukin-2 that I mentioned earlier, which is an immune stimulatory drug. So what happened to those patients? Well, 3 patients died during the immunotherapy phase. 2 died from infection. 1 died from central nervous system toxicity. And then 2 patients developed grade 2 or higher graft versus host disease, which is a known complication of transplant and usually pretty well managed. The 5-year event-free survival, so survival without relapse, and then overall survival in this group was 43% and 53% respectively. The 5-year event-free survival, again, survival without relapse, and overall survival in patients who achieved either a complete response or a partial response after transplant, but before receiving immunotherapy, was 52% and 44% respectively. So basically, roughly half of the children who achieved a good response post-transplant were alive and well 5 years later. And the authors concluded that the antibody therapy administered in this setting was perhaps more effective because it utilized the donor immune cells to mediate the anti-tumor efficacy. So I thought this data was interesting and encouraging, so I wanted to share it with you at this meeting today. And that is my last slide, so I'd be happy to answer any questions that you have.
Thank you, Dr. Desantes.
And I understand if everyone's questioned out too. Okay.
That guy who asked the question. It's okay. Thank you for the talk. I'm very curious, that second one that you mentioned, the CU sartate, is there investigation ongoing about using that as a diagnostic tool?
Just as part of this particular trial. So okay. The patients who enroll have to have the copper sartate scan before they would move on to the therapeutic phase. It's very, we've talked about using different imaging agents and trying to demonstrate, superiority, for example, to MIBG. Those kind of studies are very difficult to run because, in general, for understandable reasons, families are just not terribly interested in, having their child enrolled in that type of a study. So there hasn't been a separate trial looking at utilizing like a copper 64 diagnostic scan and comparing that to MIBG. I have one, anecdotal experience I can share with you, and that is we had a patient at our center who had disease detected in the liver based on MIBG scanning, but we couldn't see any lesions on an MRI. But there was something there on MIBG. So we enrolled the patient on this trial, and we did the copper sartate scan, and we didn't see anything in the liver. And I think that was actually helpful because I believe the MIBG scan was a false positive, and there really wasn't anything there. And this patient has continued to do well, probably a year and a half later with no evidence of disease. But there isn't a separate trial using copper 64 sartate for scanning.
Okay, it's really interesting to me because I don't want to speak for anybody else in the room, but as a father of a neuroblastoma patient, one of the things our doctors always tell us is that you never know, there might be some disease left in your child's body that we can't detect with our existing methods. And so when I saw that the copper sartate was finding stuff that the MIBG wasn't, I actually got really, really excited about that. Maybe I'm being naive here, but when I find out about another diagnostic that might do a better job of finding this damn disease in our children, I get excited about that.
Sure. And remember, this is, I'm showing you one scan on one patient, so we don't have the data yet looking at a larger patient population to be able to compare the copper sartate scans with the MIBG scans in a much larger patient population.
Okay, but you said that maybe parents wouldn't be as interested in enrolling their child on a trial that does that. Sign me up.
Okay, good to know.
Okay, we'll go over there.
Hi, thanks again for sharing. MiNiVan, how many people did you say you had enrolled? Did you reach your max enrollment on MiNiVan, or?
No, enrollment is continuing. We actually fairly recently received funding to expand the study to enroll 12 additional patients. So there have been, I think, maybe 4 patients enrolled in England. So the study is supposed to involve 3 different sites. Our site in the United States, there's a site in England, and then one in Germany that hasn't opened yet. So we need probably about 6 additional patients to be enrolled before the study will meet its accrual goals.
Okay. And then that last slide, when you mentioned the transplant and then the parent donated it, when you said post-transplant, do you just mean you're just wiping out the immune system to make room for the parent's donor cells? Or is this true, like autologous stem cell transplant, like, you know, the tandem transplants that neuroblastoma patients undergo?
Yeah, sure. Sorry, I didn't make that more clear. So it's a true transplant. It will be classified as what we would say an allogeneic transplant, where the donor donates stem cells to the patient. But in addition to stem cells, they're also donating their immune system to the patient. So the way we do this kind of a transplant is that we collect stem cells from the parent, from the donor. The patient goes through transplant-type chemotherapy. It's actually better tolerated, I will say, than what we do with autologous transplants for neuroblastoma. The treatment is known as a reduced intensity regimen, so it's not as toxic as what we see with the autologous transplants, but it still pretty much, destroys the patient's bone marrow. And then when we infuse the parent stem cells, we first remove some immune cells, those that can cause a very severe complication of transplant, which I mentioned, graft versus host disease. We take those out in the laboratory, and then we give the patient all of the parent stem cells and other immune cells, like natural killer cells we heard about, and there are other immune cells that are transferred from the parent to the patient at the time of transplant that can help mediate an anti-tumor effect. And then the stem cells ultimately establish an entirely new immune system within the patient, which is the parent's immune system.
Right. So the point of the chemo is just to make room for those parent cells. It's not
Yeah,
like the
yeah,
chemo to kill all the neuroblastoma.
Right. There may be, some anti-tumor effect of the what we call transplant conditioning regimen,
Okay.
but you're right. The main goal is to make room for the parent stem cells to grow and to provide some immune suppression to the patient so that they don't reject the parent stem cells. Because bone marrow or stem cells, it's like an organ like liver or kidney transplant. It can be rejected. So you have to give some immune suppression initially so that the new donor stem cells are accepted into the patient's body.
Okay, thank you.
Sure.
Okay, one other question there? Sure.
You actually just answered my question.
Oh.
So, it was about the toxicity of haploid versus non-haploid. So.
Yeah.
Good now.
Yeah. Okay. Yeah. We've we've treated a number of patients with this approach, and it's actually there are definitely risks involved with the transplant. I'm not saying it's not a walk in the park, but the actual conditioning regimen is pretty well tolerated. Patients usually do better than they do compared to the autologous transplant regimens that we're using for neuroblastoma today.
Okay, and that's it with the questions? Okay, thank you, Dr. Desantes.